So we are starting the chapter 12, Respiration in Plants. All of us breathe to live, but why is breathing so essential to life? What happens when we breathe?
Happy
Also,
to
do all
hear
living
that
organisms,
you are
including plants and microbes,
satisfied
breathe?
with the
If so, how?
product.
All living organisms
Your
needs energy for
feedback
carrying out
is
daily life
very
activities, be
important
it
for
absorption,
us.
transport, movement, reproduction, or
Can you
even
please
breathing.
rate our
Where does
product
all this energy
on the
comes from? We know we eat food for
Amazon app
energy,
now?
but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get
You
their
have
energy from?
to rate
And
us
microorganisms,
on the
for their energy
scale of
requirement, do
1
they
to
eat food?
5
You may wonder
stars,
at the several questions raised above.
where
They may
5
seem to be very
star is
disconnected,
being the
but in reality, the
highest.
process of breathing is very much connected to the process of release of energy from food. Let us try and understand
I
how this
will
happens.
assist
All the
you.
energy required for life process is obtained by the oxidation of some macromolecules that we
I
call
will
food.
assist
Only
you
green plants
how
and
you have
cyanobacteria
to
can
rate.
prepare their oven food by the
Can you
process of
please
photosynthesis.
open the
They trap
Amazon
light
app?
energy and convert it into chemical energy that is stored in the bonds of carbohydrates like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissues, and organs photosynthesize. Only cells containing
I'm
chloroplast
really
that are most
sorry
often
for
located in
the
the superficial layers
disturbance
carry out
in the
photosynthesis.
middle of your
सारे सेल नहीं करते हैं,
driving.
केवल वो सेल करते हैं जिनमें क्या होता है क्लोरोप्लास्ट.
May I
ठीक है.
know the right
Hence, even in green
time?
plants, all living
When
organs,
can I
tissues,
call you
and
back?
cells that are non-green need food for oxidation. Hence, food has to be translocated to all non-green parts. Animals are heterotrophic, that is, they obtain food from plants
Today
directly,
after
herbivores, or
5:30.
indirectly, carnivores. Saprophytes, like fungi, are dependent on dead
Thank you
and
for
decaying matter.
your
What is important to
information.
recognize is that
We'll connect
ultimately
you back
all the
at
food
your
that is
preferred
required for life
time for
processes
the
comes from
feedback.
photosynthesis. This
Thank
chapter
you
deals with
for your
cellular
time.
respiration, or the mechanism of breakdown of
Have a
food material
great day.
within the cell to release energy and the trapping of this energy for
Stay
synthesis of
safe.
ATP.
Have a safe
Photosynthesis of
drive
course takes
ahead.
place within the chloroplast in the eukaryotes, whereas the breakdown of complex molecules to yield,
I'm
to yield
listening
energy
to
takes
the
place in the cytoplasm and in the
podcast
mitochondria, also only in eukaryotes. The breaking of CC bond, that is carbon-carbon bonds of complex compound through oxidation within the cells, leading to release of considerable amount of energy, is called respiration. The compounds that are oxidized during this process are known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substance in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrate is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes and is trapped as a chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be, used directly but is used to synthesize ATP. which is broken down whenever and wherever energy need to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring processes of the organism, and the carbon skeleton produced during respiration is used as precursors for biosynthesis of other molecules in this cell. And the carbon skeleton produced during respiration is used as precursors for biosynthesis of other molecules in the cell. Next is, do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require oxygen for respiration to occur, and they give out CO2. Hence, plants have systems in place that ensure the availability of oxygen. Plants, unlike animals, have no specialized organs for gaseous exchange. But they have stomata and lenticels for this purpose. There are several reasons why plants can get along without respiratory organs. First, each plant part takes care of its own gas exchange needs. There is a very little transport of gases from one plant part to another. Second, plants do not present great demands for gas exchange. Roots, stems, and leaves respire at rates far lower than animals do. Only during photosynthesis are large volumes of gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. When cells photosynthesize, availability of oxygen is not a problem in these cells since oxygen is released within the cell. Third, the distance that gases must diffuse even in large, bulky plants is not great. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about thick, woody stems and roots? In stems, the living cells are organized in thin layers inside and beneath the bark. They also have openings called lenticels. The cells in the interior are dead and provide only mechanical support. Thus, most cells of the plant have at least a part of their surface in contact with air. This is also facilitated by the loose packing of parenchyma cells in leaves, stems, and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which produces CO2 and H2O as end products, yields energy, most of which is given out as heat. The equation is C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecules in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy released can be coupled to ATP synthesis. How this is done is essentially the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide, water, and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of such situations? An organism where O2 is not available? Can you think? Can you think of such situations an organism where oxygen is not available? Yes, there are many situations an organism where oxygen is not available, including anaerobic situations and anaerobic organisms. There are sufficient reasons to believe that the first cells on this planet lived in the atmosphere that lacked oxygen. Even among present day living organisms, we know of several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in others the requirement for anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. So the process next is glycolysis. The term glycolysis has originated from the Greek words glykos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Ambeden, Otto Meyerhoff, and J. Parnas, and is often referred to as EMP pathway. In anaerobic organism, it is the only process in respiration. In anaerobic organism, it is the only process in respiration, which is glycolysis. Okay. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. नींद आ रही है ना थोड़ी सी इस चैप्टर में? चलो जगाते हैं तुम्हारी नींद, ठीक है, उड़ाते हैं, आ जाओ. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of the photosynthesis. Photosynthesis का end product है sucrose, ठीक है, और from storage carbohydrate. कहाँ से मिलता है ग्लूकोज? या तो एंड प्रोडक्ट फोटोसिंथेसिस का जो की है क्या है, सुक्रोज, या फिर स्टोरेज कार्बोहाइड्रेट्स. सुक्रोज इज कन्वर्टेड इनटू ग्लूकोज एंड फ्रुक्टोज बाय द एंजाइम invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of the enzyme hexokinase. The phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure 12.1. अभी फिगर भी पढ़ रहा हूँ, ठीक है. In glycolysis, a chain of 10 reactions under the control of different enzymes take place to produce pyruvate from glucose. While studying the step of glycolysis, please note that the steps at which utilization or synthesis of ATP, or in this case NADH plus H positive takes place. H positive का मतलब बोलता हूँ मीन्स H के ऊपर प्लस, ठीक है, वो होता है. ATP is utilized at two steps. First, in the conversion of glucose into glucose 6-phosphate, and second, in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde, that is PAGAL, PGAL. We find that there is one step where NADH plus H positive is formed from NAD positive. This is when 3-phosphoglyceraldehyde PAGAL is converted to 1,3-bisphosphoglycerate, that is PPG. Two redox
calling
are removed
from
in the form of two
Eureka
hydrogen atoms
Forbes.
from
I'm
PGAL and transferred.
speaking to
I am
Jitin
repeating it again.
Goyal.
Two redox equivalents are removed in the form of two hydrogen atoms from PGAL and transferred to a molecule of NAD positive. PGAL is
Jitin
oxidized and with
Jisodiya.
inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid PGA is also an energy yielding process.
Am I audible to you?
This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Can you calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Yes, that is 2 ATP molecules are directly synthesized from one glucose molecule during glycolysis. So अब चलो steps of glycolysis पढ़ देते हैं. चलो आंखें खुली रखना मीन्स कान खुले रखना, आंखें बंद रखना. चलो स्टार्ट करते हैं. ग्लूकोज जो 6 कार्बन कंपाउंड है वहाँ पे ATP लगेगी, ATP से ADP कन्वर्ट होगा और देगा ग्लूकोज 6 फॉस्फेट. ग्लूकोज कन्वर्ट हो गया ग्लूकोज 6 फॉस्फेट जो 6 कार्बन कंपाउंड है. फिर ग्लूकोज 6 फॉस्फेट कन्वर्ट हो गया फ्रुक्टोज 6 फॉस्फेट जो कि 6 कार्बन कंपाउंड है. वहाँ पे भी ATP लगी, ATP से ADP में कन्वर्ट हो गया. फिर फ्रुक्टोज 6 फॉस्फेट कन्वर्ट हो गया फ्रुक्टोज 1,6 बिस फॉस्फेट जो कि 6 कार्बन कंपाउंड है. फ्रुक्टोज 1,6 बिस फॉस्फेट कन्वर्ट हो गया ट्रायोस फॉस्फेट में जिसको ग्लाइसेराल्डिहाइड 3 फॉस्फेट कहते हैं जो कि 3 कार्बन का कंपाउंड है. और वहाँ से फिर एक और 3 कार्बन का कंपाउंड बन गया ट्रायोस फॉस्फेट डाईहाइड्रोक्सी एसीटोन फॉस्फेट. 6 कार्बन 3 में टूट गए. ठीक है. अब ट्रायोस फॉस्फेट जो ग्लाइसेराल्डिहाइड 3 फॉस्फेट 3 कार्बन का जो कंपाउंड है वहाँ पे NAD पॉजिटिव से NADH प्लस H पॉजिटिव कन्वर्ट हो गया. NAD प्लस किसमें कन्वर्ट हो गया NADH प्लस H पॉजिटिव में. फिर वहाँ से 2 इंटू ट्रायोस बिस फॉस्फेट में कन्वर्ट हो गया ट्रायोस फॉस्फेट जो कि 1,3 बिस फॉस्फोग्लाइसरिक एसिड है 3 कार्बन कंपाउंड. और फिर 1,3 बिस फॉस्फोग्लाइसरिक एसिड इसमें कन्वर्ट हो गया 3 फॉस्फोग्लाइसरिक एसिड 2 इंटू ट्रायोस फॉस्फेट जो कि 3 कार्बन कंपाउंड है. और वहाँ पे क्या लगा है ADP लगाया जहाँ से ATP निकला है. ठीक है. ADP लगाया. ठीक है. 3 फॉस्फोग्लाइसरिक एसिड कन्वर्ट हो गया 2 फॉस्फोग्लाइसरोलेट में 2 इंटू 2 फॉस्फोग्लाइसरोलेट. और यहाँ पानी निकल गया H2O और वो कन्वर्ट होगा फॉस्फोइनॉल पायरुवेट 2 इंटू फॉस्फोइनॉल पायरुवेट और वो कन्वर्ट हो गया 2 इंटू पायरुविक एसिड क्योंकि वहाँ से 2 टर्न साइकिल चलती है. ठीक है. कहाँ से ट्रायोस फॉस्फेट से जो 3 कार्बन में टूटा है इसलिए 2 है सब क्या है. अंडरस्टूड. ADP से ATP बन गया और वो कन्वर्ट हो गया 2 फॉस्फोइनॉल पायरुवेट कन्वर्ट हो गया 2 इंटू पायरुविक एसिड जो कि 3 कार्बन कंपाउंड. Pyruvic acid is then key product of glycolysis. What is the metabolic fat of pyruvate? This depends on the cellular need. इस पर डिपेंड करता है cellular need पे. There are 3 major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic conditions in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply, that is O2 supply. Next is fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by sets of reaction where pyruvic acid is converted to CO2 and ethanol. CO2 and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organelles like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure 12.2. In animal cells also, like muscles, during exercise when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H positive, which is reoxidized to NAD positive in both the process. NAD positive का मतलब NAD के ऊपर प्लस है, ठीक है. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released, and not all of it is trapped as high energy forms of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is the number of ATPs that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis. Question again taken. So here, calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis. So the answer is 1 glucose molecule yields 4 ATP molecules in total during glycolysis. Since 2 ATP are used up in the first phase of the glycolysis, there is a net gain of 2 ATP molecules. Understood? How do you think alcohol beverages of alcohol content greater than this concentration is obtained? What then is the process by which organism can carry out complete oxidation of glucose and extract the energy stored to synthesize a larger number of ATP molecules needed for cellular metabolism? In eukaryotes, this step takes place within the mitochondria, and this requires oxygen. Aerobic respiration is the process that leads to the complete oxidation of organic substance in the presence of oxygen and releases CO2, water, and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We will look at the processes in the next section. Aerobic respiration is the process that leads to the complete oxidation of organic substance in presence of oxygen and releases CO2, water, and large amount of energy present in the substrate. This type of respiration is most common in higher organisms. Major types of anaerobic respiration सबसे ऊपर ग्लूकोज ग्लाइसरॉल्डिहाइड में 3 फॉस्फेट के वहाँ से 3 फॉस्फोग्लाइसरिक एसिड बना, फिर वहाँ से फॉस्फोग्लाइसरिक एसिड बना, और जो सेकंड स्टेप है ग्लाइसरॉल्डिहाइड 3 फॉस्फेट से वहाँ पे 3 फॉस्फोग्लाइसरिक एसिड बना, वहाँ पे NAD पॉजिटिव कन्वर्ट हो गई है NADH प्लस H पॉजिटिव, ठीक है. फर्स्ट में पायरुविक एसिड है जो वहाँ से पायरुविक एसिड में कन्वर्ट हुआ और फिर वहाँ से एथेनॉल और CO2 निकल गया. NADH पॉजिटिव H पॉजिटिव को कन्वर्ट करके NAD प्लस में कौन से स्टेप पर पायरुविक एसिड से एथेनॉल प्लस CO2 बनेंगे जब, और पायरुविक एसिड कन्वर्ट हो गया लैक्टिक एसिड में, वहाँ पे NaDH प्लस H पॉजिटिव से कन्वर्ट हो गया NAD पॉजिटिव में, जो कि पायरुविक एसिड से लैक्टिक एसिड बना, वहाँ पे NaDH प्लस H पॉजिटिव कन्वर्ट हो गया NAD पॉजिटिव में। ठीक है, नेक्स्ट इज एरोबिक रेस्पिरेशन। For aerobic respiration to takes place within the mitochondria, the final product, glycolysis, pyruvate is transported from the cytoplasm into the mitochondria. थोड़ा चैप्टर बोरिंग है इसलिए नींद आ रही होगी आपको, बट मैं सुन रहा हूँ, सो मत जाना, ठीक है। चलो, for aerobic respiration to takes place within the mitochondria, the final product of glycolysis, pyruvate, is transported from the cytoplasm into the mitochondria. This crucial event in aerobic respiration are complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms and leaving 3 molecules of CO2. The complete oxidation of pyruvate by the stepwise removal of all the hydrogen bonds, leaving 3 molecules of CO2. Next is the passing on of the electrons removed as the part of the hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. थोड़ा चैप्टर बोरिंग है, नींद आ रही होगी ना, चलो उड़ाते हैं। The passing on of the electron removed as part of the hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. What is interesting to note is that the first process takes place in the matrix of mitochondria. Consequently, the complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of CO2. The first step takes place in the matrix of mitochondria, while the second process is located on the inner membrane of the mitochondria. The second process is the passing on of the electron removed as a part of the hydrogen atom to molecular oxygen with simultaneous synthesis of ATP. Pyruvate, which is formed by the glycolytic metabolism of carbohydrate in the cytosol, after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by pyruvate dehydrogenase. The reaction catalyzed by the pyruvate dehydrogenase require dehydrogenase. The reaction catalyzed by pyruvate dehydrogenase require the participation of several coenzymes, including NAD positive and coenzyme A. The reaction is pyruvic acid plus coenzyme A plus NAD positive in the presence of Mg2 positive upon pyruvate dehydrogenase gives acetyl coenzyme A plus CO2 plus NADH plus H positive. During this process, 2 molecules of NADH are produced from the metabolism. 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid produced from 1 glucose molecule during glycolysis. The acetyl coenzyme A then enters a cyclic pathway, tricarboxylic acid cycle, more commonly called as Krebs cycle, after the scientist named Hans Krebs who first elucidated it. So the next is tricarboxylic acid cycle. The TCA cycle starts with the condensation of the acetyl group with oxaloacetic acid, that is OAA, in water to yield citric acid. and water to yield citric acid. The TCA cycle starts with the condensation of acetyl group with oxaloacetic acid, that is OAA, and water to yield citric acid. The reaction is catalyzed by the enzyme citrate synthase, and a molecule of coenzyme A, CoA, is released. Citrate is then isomerized. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation, leading to the formation of alpha ketogluteric acid and then succinyl coenzyme A. In the remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to OAA, allowing the cycle to continue. During the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. ये important line है, ये बहुत बार आई हुई है. I am repeating it again. During the conversion of the succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. This is the substrate level phosphorylation. That is SLP. In a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycle where NAD positive is reduced to NADH plus H positive, and 1 point where FAD positive is reduced to FADH2. The continued oxidation of acetyl coenzyme A via the tricarboxylic acid cycle TCA cycle requires the continued replacement of oxaloacetic acid. The first member of the cycle is the oxaloacetic acid. In addition, it also requires regeneration of NAD positive and FAD positive from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows. That is, pyruvic acid plus 4 NAD positive plus FAD positive plus 2H2O plus ADP plus PI in the presence of the mitochondrial matrix gives 3CO2 plus 4 NADH plus 4 H positive plus FADH2 plus ATP. I am repeating it again. Pyruvic acid plus 4 NADP NAD positive plus FAD positive plus 2H2O plus ADP plus PI in the presence of mitochondrial matrix gives 3CO2 plus 4 NADH plus H positive plus FADH2 plus ATP. सिट्रिक एसिड साइकिल की वो जो चार्ट है वो पढ़ रहा हूँ. चलो ठीक है. पायरुवेट है 3 कार्बन कंपाउंड, वहाँ पे कोएंजाइम ए NAD प्लस कन्वर्ट हो गया NADH प्लस H पॉजिटिव, वहाँ से CO2 निकल गया. फिर वो एसिटाइल कोएंजाइम जो 2 कार्बन कंपाउंड है वहाँ से साइकिल स्टार्ट हुई. सिट्रिक एसिड जो 6 कार्बन कंपाउंड है वहाँ से CO2 निकल गया, NAD पॉजिटिव NADH प्लस H पॉजिटिव में कन्वर्ट हो गया. फिर सिट्रिक एसिड अल्फा कीटोग्लूटेरिक एसिड में कन्वर्ट हो गया जो कि 5 कार्बन कंपाउंड है, वहाँ से CO2 बाहर निकल गई. NAD से NADH प्लस H पॉजिटिव कन्वर्ट हो गया और GDP से GTP बन गया. फिर आ गया सक्सिनिक एसिड जो कि 4 कार्बन कंपाउंड है. फिर सक्सिनिक कन्वर्ट हो गया मालिक एसिड में जो 4 कार्बन कंपाउंड है, और यहाँ पर FAD से FADH2 कन्वर्ट हो गया. FAD प्लस कन्वर्ट हो गया FADH2. मालिक एसिड कन्वर्ट हो गया ऑक्सलोएसिटिक एसिड में जो कि 4 कार्बन कंपाउंड है. इस स्टेप पर NAD पॉजिटिव कन्वर्ट हो गया NADH प्लस H पॉजिटिव में. क्लियर? ठीक है. अब नहीं समझ आया होगा तो डायग्राम निकाल के देख लेना एक बार. ठीक है. We have till now seen that glucose has been broken down to release CO2 and 8 molecules of NADH plus H positive, 2 of FADH2 have been synthesized beside just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither oxygen has come into the picture, nor the promised large number of ATP has yet been synthesized. Also, what is the role of the NADH plus H positive and FADH2 that is synthesized? Let us now understand the role of oxygen in respiration and how ATP is synthesized. So the next is ETS, that is electron transport system, and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H positive and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to O2, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport system, that is ETS, and it is present in the inner mitochondrial membrane. ETS कहाँ पे present होता है? Inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by an NADH dehydrogenase, that is complex first, and electrons are then transferred to the ubiquinone located within the inner membrane. Ubiquinone also receive reducing equivalents via FADH2, that is complex 2, that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinone is then oxidized with the transfer of electron to cytochrome C via cytochrome BC1 complex, that is complex 3rd. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electron between complex 3rd and 4th. Cytochrome C क्या काम कर रहा है? Mobile carrier for transfer of electron between complex 3rd and 4th. Complex 4th refers to cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper centers. Complex 4 refers to cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper centers. When the electrons pass from one carrier to another via complex 1st to 4th in the electron transport chain, they are coupled to ATP synthase, that is complex 5th, for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor. Unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation. In respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane-linked ATP synthesis as explained by chemoosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase. Complex 5th. This complex consists of two major components, F1 and F0. The F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein that forms the channel through which proton cross the inner membrane. F0 is an integral membrane protein complex that forms the channel through which proton cross the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. The passage of proton through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 4 hydrogen positive passes through F0 from the intermembrane space to the matrix down the electrochemical proton gradient. So now I am going to read the electron transport system का जो diagram है यहाँ पे दे रखा है intermembrane space, inner mitochondrial membrane और matrix. First NADH plus H पॉजिटिव कन्वर्ट हुआ NAD पॉजिटिव में जो कि कॉम्प्लेक्स फर्स्ट है NADH डिहाइड्रोजनेज, कॉम्प्लेक्स थर्ड साइटोक्रोम BC1 है, कॉम्प्लेक्स सेकंड सक्सिनेट डिहाइड्रोजनेज, कॉम्प्लेक्स फोर्थ साइटोक्रोम C ऑक्सीडेज. वैसे ये डायग्राम इतना इंपॉर्टेंट नहीं है NCERT में जो दिया हुआ ETS का. जो दिया हुआ है डायग्रामेटिक प्रेजेंटेशन ऑफ एटीपी सिंथेसिस इन माइटोकॉन्ड्रिया. आउटर साइड दे रखी है, इनर माइटोकॉन्ड्रियल मेम्ब्रेन दे रखी है और मैट्रिक्स दिया हुआ है. F0 आउटर साइड पे बीच में है और 2H पॉजिटिव ATP और PI को कन्वर्ट कर रहा है ATP में. द रेस्पिरेटरी बैलेंस शीट. It is possible to make calculation of the net gain of the ATP for every glucose molecule oxidized, but in reality this can remain only a theoretical exercise. This calculation can be made only on certain assumptions that there is a sequential orderly pathway functioning with one substrate forming the next and with glycolysis, TCA cycle and ETS pathway following one after another. The NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of the intermediate in the pathway are utilized to synthesize any other compound. Only glucose is being respired. No other alternative substrate are entering in the pathway at any of the intermediary stages. But these kind of assumptions are not really valid in a living system. All pathway works simultaneously and do not take place one after another. Substrates enter the pathway and are withdrawn from it as and when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means, yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, there can be a net gain of 38 ATP molecules during aerobic respiration of 1 molecule of glucose. Now, let us compare fermentation and aerobic respiration. इसको कंपेयर कर रहे हैं फर्मेंटेशन को और एरोबिक respiration. So let's start the comparison. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to CO2 and H2O. In fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions. Next is NADH is oxidized to NAD positive rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. Next is amphibolic pathway. Glucose is the favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. It's a true statement or false statement. It's true statement. ठीक है? Repeating the line again. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrates can also be respired as has been mentioned earlier, but they do not enter the respiratory pathway at the first step. See figure 12.6 to see the points of entry of different substrates in a respiratory pathway. Fats would need to be broken down into glycerol and fatty acid first. If fatty acid were to be respired, they would first to be degraded to acetyl coenzyme and enter the pathway. Glycerol would enter the pathway after being converted to PGAL. Glycerol कब आएगा जब वो PGAL में convert होगा. Protein would be degraded by proteases. किससे होंगे? Proteases. And the individual amino acid after deamination, depending on their structure, would enter the pathway at some stages within the Krebs cycle or even as pyruvate or acetyl coenzyme. Since respiration involves breakdown of substrate, the respiratory process has traditionally been considered as catabolic process and the respiratory pathway as a catabolic pathway. But is this understanding correct? We have discussed above at which points in the respiratory pathway different substrate would enter if they were to be respired and used to derive energy. What is important to recognize is that it is this very compound that would be withdrawn from the respiratory pathway for the synthesis of the said substrates. Hence, fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as a substrate. Fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as substrate. But when the organism need to synthesize fatty acid, acetyl coenzyme would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into the picture both during the breakdown and synthesis of fatty acid. Similarly, during breakdown and synthesis of protein 2, respiratory intermediate form the link. Breaking down process within the living organism is catabolism and synthesis is anabolism because the respiratory pathway is involved in both anabolism and catabolism. It would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one. जो चार्ट दी हुई है चलो वो पढ़ते हैं. यहाँ पे फैट्स, कार्बोहाइड्रेट और प्रोटीन दिए हुए हैं. फैट्स ब्रेकन होगा फैटी एसिड और ग्लाइसरॉल में, फिर वो कन्वर्ट होगा एसिटाइल कोएंजाइम में, फिर वो जाएगा क्रेब्स साइकिल में, वहाँ से H2 निकल गया, CO2 निकल गया. कार्बोहाइड्रेट कन्वर्ट होंगे फर्स्ट सिंपल शुगर्स में, एग्जांपल ग्लूकोज 6 फॉस्फेट, फिर फ्रुक्टोज 1,6 बिस्फॉट में जाएगा, फिर ड्राई हाइड्रोक्सी एसीटोन फॉस्फेट में जाएंगे, ग्लाइसरॉल्डिहाइड 3 फॉस्फेट, फिर वहाँ से पायरुविक एसिड में आएंगे और फिर एसिटाइल कोएंजाइम में आएंगे. ठीक है. और प्रोटीन होंगे अमीनो एसिड में कन्वर्ट, फिर वो जाएंगे नीचे पायरुविक एसिड में, फिर वहाँ से एसिटाइल कोएंजाइम में आएंगे और फिर क्रेब्स साइकिल में आएंगे। ठीक है, ये था इंटर रिलेशनशिप अमंग द मेटाबॉलिक पाथवे शोइंग रेस्पिरेशन मीडिएटेड ब्रेकडाउन ऑफ डिफरेंट ऑर्गेनिक मॉलिक्यूल्स to CO2 and H2O. Next is respiratory quotient. Let us now look at another aspect of respiration. As you know, during aerobic respiration, O2 is consumed and CO2 is released. The ratio of the volume of CO2 evolved to the volume of O2 consumed in respiration is called the respiratory quotient or respiratory ratio. RQ is equal to volume of CO2 evolved upon volume of oxygen consumed. Volume of CO2 evolved upon volume of O2 consumed. The respiratory quotient depends upon the type
Good
of respiratory substrate
afternoon.
used during respiration. When carbohydrates are used as substrate and are completely
This
oxidized,
is
the
service
RQ will
call.
be 1
I
because
am
equal amount of
Kritik
CO2 and
calling
O2 are
from
evolved and
Eureka
consumed
folks.
respectively, as shown in the
I'm
equation below.
speaking to
That is 6
Chetan
C6H12
Tingase.
O6 C6H12O6 plus 6 O2 gives
It's a
6 CO2 plus
feedback
6
call
H12 plus energy.
regarding the
RQ value is equal
aquaguard
to 6
for the
CO2 upon
light
6 O2 is equal to 1.
which you have
When
recently
fats are used in
purchased
respiration,
from
the
Amazon app.
RQ is less than 1.
Am I right?
Calculation for a fatty acid tripalmitin if used as substrate is shown.
I
That is
hope the
2C51
installation has been successfully
H98O6 plus
completed.
145 O2 gives 102 CO2 plus 98 H2O plus energy. ट्राईपाल्मिटीन का है
And the
C51
product is now
H98O6.
absolutely
RQ
working
value is equal to
fine.
102 CO2 upon 145 O2 is equal to 0.7. ट्राईपाल्मिटीन की रेस्पिरेटरी क्वोशिएंट वैल्यू RQ. When protein are respiratory
Are you
substrate, the ratio would be
satisfied
about
with
0.9.
our product,
What is important to
Chetanji?
recognize is that in living organisms, respiratory substrate are often more than one. Pure proteins or fats are never used as substrate
We
as
are
respiratory
happy to
substrate.
hear that you are
Pure
satisfied
protein or
with the
fats are never
product.
used as respiratory substrates.
Your
Summary.
feedback
चलो,
is
इसको
very
read लेते हैं.
important for
Plants,
us.
unlike animals, have no
Can
special
you please
system for netting or
rate
capsule
our
exchange.
product
Stomata and
on the
lenticels allow gaseous
Amazon
exchange by
app
diffusion.
now?
Almost all living cells in plant have their surfaces exposed to air. Almost all living cells in plant have their surface exposed to air. The breaking of CC bond of complex organic
You
molecule by
have to
oxidation cells
rate us
leading to the
on
release
the
of a
scale of
lot of
1
energy
to
is
5
called cellular
star,
respiration.
where 5
Glucose is the
star
preferred
is being
substrate
the
for respiration.
highest.
Fats and protein can also be
It
broken down
will just
to yield
take
energy.
less than minute
The
of
initial
your
stage of
time,
cellular respiration takes
and
place in
I
the
will
cytoplasm.
assist you how
Each
you
glucose
have to
molecule is
rate.
broken through a series of
Can you
enzymes
please
catalyzed
open the
reaction into two
Amazon
molecules
app
of
now,
pyruvic acid.
Chetanji?
This process is called glycolysis. The fat of the pyruvate depends on the availability of the oxygen in the organism. Under anaerobic condition, either lactic acid fermentation or alcohol fermentation occurs. Fermentation takes place under anaerobic conditions in many prokaryotes, unicellular eukaryotes, and
May
in
I know
germinating
the
cells.
right
In
time?
eukaryotic organisms, aerobic
When
respiration
can
occurs in
I
the
call
presence of
you
oxygen.
back for your
Pyruvic acid is transported
valuable
into the mitochondria
feedback
where it is converted
confirmation,
into acetyl coenzyme
Chetanji?
with the release of CO2. Acetyl coenzyme then enters the tricarboxylic acid pathway or Krebs cycle operating in the matrix of the
Today
mitochondria.
after
NADH
6
plus H
p.m.
positive and FADH2 are generated in the Krebs cycles. The energy in these molecules as well as that in the
And if
NADH
you are
plus H positive synthesized
comfortable in
during
sharing
glycolysis
it now, we
are
can
used for
register
synthesis.
your
Moringa are
valuable
used to synthesize
feedback
ATP.
here.
The energy in this
so
molecule as well
that
as
you
that in the
will not
NADH
get
plus H
any
positive
further
subunits during
follow-up
glycolysis are
calls.
used to synthesize
Are you
ATP.
comfortable
This
in
is
sharing
accomplished
it
through a
now or
system of electron
later?
carriers called electron transport system ETS, located on the inner membrane of the mitochondria. The electron, as they move through the system, release enough energy that are
Are you
trapped to synthesize ATP.
Are you
This is called
comfortable in
oxidative
sharing
phosphorylation.
it now or
In this
later?
process, O2 is the ultimate acceptor of electron and it gets reduced to water. In this process, O2 is the ultimate acceptor
Surely we
of
will
electron and
connect you back at your
it gets
preferred time
reduced to
for
water.
the feedback
The respiratory pathway
confirmation.
is an amphibolic pathway
Thank you
as it
for being
involves
the
both
part of the
anabolism and
Eureka
catabolism.
Fox family.
The respiratory quotient
Stay safe,
depend upon the type of
stay
respiratory
healthy,
substrate used during
have
respiration.
a great
With this,
day.
complete the chapter. Thank you for hearing.
समय
एक
देने
बार
के
और
लिए
सुन लेना,
आपका बहुत
थोड़ा बोरिंग
बहुत
चैप्टर है।
धन्यवाद। आपका दिन शुभ रहे। थैंक